Hello. We will just continue the same nickel enzymes what we are discussing last time. So, in this nickel enzyme, we are focusing our attention mostly on nickel and its coordination chemistry, and slowly we can move to some complex species which is based on nickel, iron and sulfur. So, this can also be classified as nickel, iron, sulfur proteins. So, which will be definitely a different one if we just compare with that with what we know as the corresponding iron sulfur proteins. So, these we know that there are some nickel enzymes which are very much useful in metabolism of chemicals which are abundant in the pre-oxygen evolutionary era. So, we want to assimilate some of these species like methane, whether we can produce it or we can get some species where the methyl group is attached to the nickel center, then carbon monoxide and hydrogen. So, these has some similarity with that of our organometallic chemistry, where we can have direct nickel carbon bond and you can something where we can make some nickel methyl bond or nickel carbonyl bond or nickel hydride or some other species related to hydrogen. And all these species are mostly they present in very high level in anaerobic bacteria. That means, when we do not have in the environment dioxygen, so environment is fully populated with the gases like methane, carbon monoxide and hydrogen. And if we just compare the amount of that nickel what we find in our system, that means the mammalian serum, which is simply 100-fold excess than the concentration what we require for iron in our blood and any other important species like ferritin or transferrin, zinc in the different proteins and the enzymes and the copper. So, these are the three most abundant which are 100-fold excess than nickel. So, nickel concentration is very less, but still nickel can function very easily due to its characteristic property related to its coordination chemistry or biocoordination chemistry, which we cannot get for iron center or zinc center and copper center. So, what is that? So, the stabilization of nickel in different oxidation states. We all know that the nickel is mostly stabilized in +2 oxidation states. So, most of the compounds where we get in the laboratory also the coordination chemistry of nickel is mostly dominated by nickel 2 plus, and what we have seen earlier also in case of urease, that is the hydrolytic species which is responsible for hydrolysis of the urea molecule, and the coordination chemistry is mostly based on a species which is 3D8 in different coordination geometries. So, first time we will focusing our attention on nickel in lower oxidation state. So, you have large number of 3D electrons, already you have a 3D8 system in +2, and if we go to nickel 1, so there will be more than that species, that means 3D9 system, and electrons are forced into exposed sigma and pi orbitals. So, those electrons, the available electrons on those sigma and pi electrons would be responsible for very different or characteristic property what you can get based on nickel. So, not only the lower oxidation state, that means nickel in +1 oxidation state, but also the tetragonal oxidation state, that means the nickel 3, which can give rise to some reactive free radical and which is also very much useful, like the if you can recall the corresponding organometallic chemistry based on palladium. So, palladium we all know we start from palladium 2, then if we are able to reduce it to palladium 1, then immediately 2 electron transfer can transfer that particular oxidation state from +1 to +3. And not only this one, but also something else, that means when we have a nickel in +2 and if we can reduce it by 1 electron, so you get a nickel 1 plus species. So, this particular species, so in this particular case you have a coordination geometry which will be very much similar to that of a square planar geometry. But if you go for a corresponding oxidized form, that means nickel in 3 plus. So, it will have a general tendency to form a corresponding octahedral species. So, not only the electron transfer, because all these cases what we will find for hydrogenases now. So, you will be getting something where you have the corresponding assimilation of H2 molecule, or you are producing that H2 from proton. So, you can sometime you accommodate, that means the expansion in the coordination number as well as transfer of electron. So, if you have 2 such H plus, so you require 2 electron for that transformation of H plus to H2. So, nickel is ideally suited for that if you have some stable oxidation state. So, this is the most stable oxidation state, and this is the catalytically active one, and this also is the catalytically active oxidation state, and it can change the corresponding coordination number also, like that of our organometallic transformations based on palladium, that when you go to +3 oxidation state, it can attract 2 more coordination sites, such that if you have H base some species, that means a hydride or H plus or other species something like that of your solvent. So, it can occupy this site as well as it can occupy this site. So, this tetragonal nickel 3, which can also give rise to some reactive free radical, is also important to know when we talk about this thing. So, if you go for some nickel iron sulfur species, this is very much unusual, which is very difficult to make also. So, if we want to make it in as a model compound in the laboratory, which is also very difficult to make, that means what we are talking about something where you have a nickel iron binuclear compound, because they have some different preference for oxidation state, and sometimes when you go for this nickel and iron species, there must be something, that means there must be some bridging unit, and that bridging unit should have some preference to bind this both nickel and iron. So, it is definitely a very unusual site, and that site what we get in case of this particular reactivity pattern. So, initially we can have some of these enzymes where nickel appears to be selective in its plasticity of coordination and redox chemistry, that just now I am talking about, that you can move from one coordination site to another coordination site. That means, you can move from a square planar geometry to the octahedral geometry. So, that is why we are talking it as a plasticity in the coordination geometry as well as the redox chemistry. That means, how much your oxidation states are accessible to you to a biologically feasible oxidizing agent or reducing agent. You cannot use some other strong chemical reagent, chemical oxidizing agent or reducing agent to get some reduced form or the oxidized form. And when it can cycle through the 3 different oxidation states, that means, when you start from the most stable oxidation state, which may not be catalytically active, but you can go down to +1 oxidation state and you can go up to +3 oxidation state, and it can catalyze large number of reactions within a potential window of 1.5 volt. So, we are not going much for this oxidation and the reduction potential. So, you have a window for +1 oxidation state to +3 oxidation state, which lies between 1.5 volt. And one such example, though we will not discuss in detail all these, is the superoxide dismutase family of molecules, which we know that generates O2, where the metal center can be able to show redox processes, where you have the potential. You see the window is very large window. So, +890 to -160 millivolt. So, between this window and between 1.5 volt window, we see some other examples. For one such example we will find is the carbon monoxide dehydrogenase, and they are interrelated. So, one is that carbon monoxide dehydrogenation reaction is nothing but we abbreviate it as CODH. So, is nothing but is corresponding transformation from carbon monoxide to carbon dioxide and vice versa. And another is methyl coenzyme M reductase, which is also possible for the reduction reaction. And again, if we talk in terms of the corresponding potential values like this and the superoxide dismutase, and for this it is a quite negative potential, which is minus 600 millivolt. So, you require some strong reducing agent to generate a species which can be nickel in +1 oxidation state, and that +1 oxidation state, if you are able to generate, that can show some catalytic activity. So, the potential is important as well as your corresponding coordination geometry. So, when you go for these hydrogenases, and these hydrogenases, one important thing will be you get the binuclear species based on nickel and iron, and some bridging group is there, and another bridging group is there, and we have 2 other coordination site and 3 more coordination sites. So, the analysis initially established that you have cysteine sulfur residues or selenocysteine residues, selenium based cysteine, sulfur is replaced by selenium. So, if we can have all these groups, and these we know that cysteine residues are based on S minus. So, this can very well used as a bridging unit between nickel and iron, and these are coming from protein chains. So, these are protein ligands. So, if you have all 4, that means, if these are also cysteine sulfur, this is also cysteine sulfur. So, what you need? You need 4 cysteine sulfur groups around a nickel center. So, that is also very important if you are able to make in the laboratory that some compound where you have the environment is simply NiS4, and that S4 at one side, if they are all thiolate groups, that means the thiol ligands are very good ligand for bridging the second group. So, already you have the mononuclear nickel center, and 2 of these, that means, if it is the different geometries, not a square planar one, this can be a typical square planar geometry. But if you have available thiolate units from these 2 sides, and if they are used to bridge the second metal, you get something where you can give a heterodinuclear system. So, this is one such strategy, that you do not use anything exogenous, that means, which is not present within the protein chain or within the biological system, but it is already bound to the nickel center. So, this nickel is nothing but your nickel cysteine sulfur 4, which is equivalent to that of our well known iron site rubidoxine. This we all know. So, rubidoxine is also a mononuclear site where 4 cysteine sulfur residues are attached to your iron site. But in this particular case, if something happens like this, that if you bring the second center, that means the iron center, with some of the ligands like this ligand and this ligand, say they are in a facial orientation. So, already we know that anything in a face, basically some well known ligand, we know that the triazacyclanone type of ligand. So, one face is occupied. So, basically if you have some species like that in the laboratory also you can make. one ligand L, which is like a tridentate one, and that tridentate one is capping one face of the iron, and you can get a corresponding salt which is chloride salt. So, this iron salt you can make in the laboratory a synthetic molecule of this type, which is 5 coordinated. So, now if you put something, that means if you are able to make some of this type of compound, that how you attach this particular unit where you have 4 cysteine sulfurs are already attached to the nickel side, and you have this particular unit 5 coordinated side. So, what you can do, you just, if you are able to remove these chlorides from iron side, and if this cysteine sulfur can bridge your iron side, so you get some assembly which will be very much similar to this. And in this hydrogenase, this particular motif is present, that means you have already, you can make this in a 5 coordinated species based on iron, which is allowed to react in the protein chain with the nickel site, which is already occupied by 4 cysteine sulfur residues, and you get some interaction such that you bring the iron side to the nickel. It is not that these hydrogenase can react in a different fashion when both of them are iron or both of them are nickel, but you need iron, because the coordination preference for this iron is completely different compared to your nickel site. So, if you have a heterobinuclear system, the two catalytic sites, the metal based catalytic sites, one is based on iron and another is based on nickel, would be completely different. So, that we get for the hydrogenases, and all known hydrogenases contain these iron sulfur clusters, and many but not all have found to contain nickel as well. So, basic thing is that you have most of these hydrogenases, you should have these iron sulfur clusters, and this iron sulfur cluster is already present there, and this iron sulfur cluster we can have, and in this particular case we are considering this as you have, this is also cysteine sulfur, this is cysteine sulfur, and these two cysteine sulfurs are the bridging cysteine sulfur residues, and which is attaching to another site which is iron, but you can now have, instead of a facially capping tridentate triazocyclononane type of ligand, you have 3 inorganic ligands, 2 cyano and 1 carbonyl. So, these 3 groups basically can stabilize this particular iron site in low oxidation state, and that was the strategy for the biological system, that you should stabilize this particular iron site in a low oxidation state, that means the +2 oxidation state. And in electron transfer reactions, because these hydrogenases are all electron transfer proteins, so they are basically electron transfer enzymes, but electron transfer will take place based on iron and these corresponding proton or the hydride units, which is coming from the cleavage of the heterolytic cleavage of the H2 molecule, but your iron site is not participating in any such electron transfer reaction. So, this H2 molecule can be cleaved heterolytically, and spectroscopic studies reveal a nickel 3 hydrido species. So, that people have identified that once you have, and we all know that the most important spectroscopic technique for identifying a nickel site is the EPR, the electron paramagnetic resonance. So, electron paramagnetic resonance once you have, which cannot identify very easily the nickel in +2 oxidation state, but it can easily identify nickel in +1 oxidation state and nickel in +3 oxidation state. So, these two, that means whether your nickel center is settling between +1 or +3 oxidation state, that can be very easily identified by EPR spectroscopic measurements, because these two species, these two oxidation states can have characteristic spectral feature, that means it has the corresponding, whether it can be a rhombic one or a monoclinic one. So, say axial one or a rhombic one, the spectral feature for the EPR, And when nickel 3 is there, you can find that in the catalytic cycle, when you will find that the hydridor species, that means when you have this nickel, we will find in the entire catalytic cycle also that you have this particular. So, nickel center oxidation state of the nickel you can identify, that means the nickel center is in +3, but the nature of this species, that means whether is a hydrogen atom or H plus or H minus, that means it is stabilized by H minus. So, this particular assembly, that means is NiH, so nickel 3 hydridor species, which is the most characteristic species which is present in hydrogenase. So, you have H2 which is attached to this nickel and it clipped between H minus and H plus, and one particular center, that means the metal center is utilized for taking up this H minus, and when H minus is taking up, it can also, if your these two distance, because already you have the bridging sulfur groups. So, we all know that if you have a single bridging unit between two metal centers, you have a corresponding distance of nickel iron. If you go for a second bridging, your distance is less. If you go for a third bridging, your distance is further less, because when we find that if your two sides are octahedral in geometry, you can have a corresponding face. So, one face of one metal center and another face of the second metal center are bridged. So, when it is triply bridged by nuclear unit, so this hydride is bridged, this sulfur is bridged and second sulfur is also bridged, you get the shortest possible nickel iron distance. So, that distance is also very important for its catalytic activity and how you modulate these two distances. And interestingly, when the cysteine sulfur is attached to nickel side, you can have something, that means this sulfur, when it is attached to the nickel side, it can bridge the second metal center. Similarly, when this sulfur is already coordinated to your nickel side, it can bind a proton like this, that means already if you have S minus which can be bound to your nickel side, or SH in the neutral form, SH in neutral form can also loosely bound to the nickel side. So, either the protonated form of cysteine sulfur or the deprotonated form of the cysteine sulfur can bind to the nickel side, and that basically can give some interesting feature, that when it is protonated, what happens? Your nickel sulfur distance will increase. It will show some loose coordination to your nickel side, like binding of water molecule to any metal center compared to your hydroxide binding. We know that the hydroxide binding is stronger to any metal center compared to your neutral water molecule. So, this binding, so binding interaction will still be there, but you have a longer distance and which is weakly bound. So, what we are basically generating? We are basically generating two sides, and for heterolytic cleavage of the H2 molecule giving H minus and H plus. So, nickel site will be available for binding H minus, and your ligand site, that means the sulfur site from the cysteine sulfur will be available for binding your proton. So, that basically gives some important reactions based on all these nickel iron hydrogenases. So, this class of molecules basically we will call them as, because there are several only iron hydrogenases, but these we will call as nickel iron hydrogenases, and they are associated to the membrane. So, they are membrane associated protein molecules, and that basically handling or taking care of proton. So, it has something related to that. We will later on we will see something related to what we basically consider as a proton pump. So, if membrane is associated and that membrane is there which can reversibly bind the proton into the system. So, because we are talking something related to the assimilation of the proton and the production of H2 for these hydrogenase reactions. So, there are basically 4 such classes of nickel iron hydrogenases, and one of them is membrane associated. So, when it is membrane associated, we call them as it is related to something which is considered as a proton pumping mechanism and energy coupling complex, because when you know that there is some proton gradient is formed from one side of the membrane to the other side, we can have some good free energy change and which will be utilized for ATP synthesis. So, in all these cases, these are a little bit complicated. So, we get some part, one such part or one such unit which we will consider as subunits. So, one subunit we consider it as a large subunit and another subunit we consider as a small subunit, and the large subunit has a molecular weight of 60 kilodalton and small is further small. And already we have seen that how a binuclear nickel iron system is important. So, you have identified it, that from the analysis also, that identification of the metal sites, that means you have iron and the nickel sites, so active sites. So, that is coupled to a wire within the 30 kilodalton small subunit. So, if you have one such unit and another one such unit, and in between you see the total thing, that means only one part we are discussing which is based on your nickel and iron. So, one is large now and another is small, and these two units are connected by further are your iron sulfur clusters, because all we know that the use of these iron sulfur clusters, because they can supply electron, they can donate electron to the system or they can take off electron from the system. So, if you have, so all these hydrogenases are your ferredoxin or rubidoxin dependent, that means the iron sulfur protein dependent. So, number of electrons supplied to the proton will be dependent on the number of these iron sulfur clusters which are present, and sometimes they are in the oxidized form, they take off electron from other biological reducing agents like NADP and others, and they will be in the reduced form, and those reduced form will be in the near vicinity of the nickel iron based hydrogenases, and those electrons will be supplied to proton for the production of hydrogen. So, if one such this thing, that means the subunit of this nickel iron center contains carbon monoxide ligand, that we have identified, which is very important, and that we will find how nature has devised something. That means, not only we are identifying the presence of nickel, the presence of iron, but also we are identifying the two different inorganic ligands, that means the carbon monoxide and cyanide ligands attached to the iron side. But this nickel and iron based hydrogenases can also tell us something, that though this carbon monoxide is attached to the iron side, but we can have something, that means this biological site or any other biological site, you can have some mechanism where the nickel center, the nickel can function as an active site which can go for assimilation of carbon monoxide. So, that gives us some clue that for carbon monoxide dehydrogenases also, not only hydrogenases, but carbon monoxide dehydrogenases which we are considering at CODH. So, those CODH can also have something to play in this system, that it can go for binding carbon monoxide in a transient form, that means it can transfer this carbon monoxide to some other species, and it can activate this carbon monoxide either in the form of transferring it from carbon dioxide to carbon monoxide or carbon monoxide to carbon dioxide. And when it is attached to the iron side, already I told you that it is stabilizing the low oxidation state, that means the ferrous state. So, this geometry already the diagram I showed you. So, this thing is that how this the large subunit and the small subunit. So, this is the thing, that means you have coverage of these, that means it is under a 30 angstroms surface of the protein, and under that it is there. So, you have to cross this particular distance, that means hydrogen is a very small molecule, and that molecule basically tunneling and going through this particular part and approaching this particular binuclear active site. So, if it is fully covered, and if say something is membrane bound thing, and if you consider that membrane is not allowing for the gas permeation. So, permeability of the hydrogen is very important there, and that permeation is allowing that particular membrane, that hydrogen is entering there and it is passing or crossing that protein sheath basically, or protein coverage, and then ultimately approaching your catalytically active site. That means, it is approaching basically to your binuclear active site. So, this already I told you that you have one iron side, the carbon monoxide and the cyanide groups are attached, and these two cysteine sulfates are the bridging units, and these two are can be cysteine and sometimes it can be also selenocysteine. So, this active site you can have now. So, how you go for this mechanism? So, mechanism is a little bit complicated one, and you have this unit because we are talking here. Your subunit is like this. On the iron, on one side you have these groups, and on the left you have the nickel and attached to your cysteine sulfate. So, what basically we are getting there, that you have some amount of vacancy here. It is not triply bridged binuclear unit. So, if you just able to consider it, that okay, this is not our system where you have two metal centers which are triply bridged. So, it is not a triply bridged system. So, if you get the introduction of hydrogen, as for the heterolytic cleavage already I showed you that you have the hydride function, the red hydride group is bridging these two metal centers, and the second hydrogen which is coming out as the proton, which is attaching to your cysteine sulfur. So, this cysteine sulfur is getting protonated. So, this cysteine sulfur is getting protonated, and when you have, so what is the driving force? So, you have this one, then H2 is attached to this particular form, and if you take out this particular H, and this particular one is the environment is such that, that means the when people discovered say 20 or 25 years back this particular system, initially they were puzzled to know that how in this particular environment, that means the nickel is basically in a S4 environment, 4 sulfur environment. So, this 4 sulfur nickel environment, how immediately from this particular step, that means your proton is moving from this cysteine sulfur to some nearby base, this is the base basically. So, nearby base in the protein envelope. So, that base is taking up this proton, and your nickel center is responsible for oxidation. So, basically what we are talking about, we are talking something related to your proton coupled electron transfer. So, this has the uniqueness for this thing, that means sometimes we consider only electron transfer, that means those metalloenzymes or the protein systems you can have only electron transfer, and some other time we will find that you can have the proton transfer from one side to the other. Sometime you can find it for the different hydrolases, but in this particular case you have the proton transfer as well as your electron transfer. So, once it is going there, so your nickel center is basically oxidized, and this particular active state, that means is the starred form. So, all these starred forms are EPR active. So, this would be EPR active. So, people have identified initially that the spectroscopic signature what you can have, that the catalytically active site is EPR active. So, this particular step you go for the activation of the active site, activation of the binuclear site, but the real catalytic cycle is this 4. So, this is the catalytic site. So, you get the nickel in +3 oxidation state, and when it moves, that means if this hydride function, that means the hydride group is moving and your nickel center is now getting reduced, that means the hydride is transferring electron. So, hydride is transferring electron. So, hydride was H minus, it is going to H plus immediately. So, it is a 2 electron step. So, those 2 electrons are donated to the nickel 3 plus oxidation state to reduce it to nickel 1. So, these two are basically, people initially were puzzled, people could not identify it for several years, that which particular site is responsible for the catalysis of hydrogenases, whether it is a trivalent nickel state or the monovalent nickel state, but both of them are EPR active, but the nature of the EPR spectrum are different. So, that we should know that the EPR spectrum for the monovalent nickel is completely different to that of your trivalent nickel. So, trivalent nickel is simply most of the cases your typical axial and rhombic signal what we find for the copper, which is a 3D9 system. So, this is your 3D7 system and this is 3D9 system. So, this can be very much similar to that of your copper system. So, if these two are different, then people can propose that these two things, that means the hydride transfer is taking place and your site is moving from a trivalent site to a monovalent site. And this particular site, again if you go for this, that means the oxidative addition, which we all know from the organometallic chemistry or anything, what we know that nickel can settle between the two oxidation state, what we all know for the palladium chemistry, that if you can have a trivalent nickel and the monovalent nickel, and the palladium chemistry is well known to you, that when we go from a trivalent state to a monovalent state, not only you are transferring electron, but also you have to move the ligand from the nickel side, because the nickel will have the preference for low coordination number. So, it cannot accommodate a coordination number of 5 or 6, it will always try to prefer a corresponding coordination number of 4. So, when you go for oxidative addition on this monovalent nickel, immediately it can go again to a trivalent one, but this particular function is a different one. Now, you have 3 H, already you have the protonated thiolate function, plus you can have this type of arrangement, that means the hydride as well as these two groups already attached to the nickel. So, this is one such proposal for that is already in your trivalent state, but this is completely different from NiA C star, NiA R star, this is NiA X star. So, these two are different, C star and X star are different. So, you will have again some signature for the trivalent nickel, but your attachment for the hydrogens are different, and this particular case you have single electron transfer over here, you can have one more single electron transfer like this, that means you have the proton transfer as well as electron transfer from this. Again you take out this particular form of this hydrogen. So, all involvement for this, that means you are basically assimilating hydrogen involving proton and the two oxidation state, that means the +1 oxidation state of nickel and +3 oxidation state. So, like your catalysis based on palladium, what we find occasionally in organometallic chemistry, that we go for some catalysis based on palladium. Similarly, in hydrogenases, which is the simplest possible catalysis for hydrogen, that you involve these two oxidation state, that means +1 and +3, but now it is based on nickel +1, and this particular one is giving you some attachment to your iron side. So, what we basically find is just you have written in the language that require activation initially involving prolonged treatment of H2 to generate NiACA star step, and replacement of some of the OH ligand if it is not at all vacant. That means, if you have a vacancy what is attached to hydride bridge, that means if we go for this, the corresponding water molecule is present, and sometime depending upon the pH of the medium you can have some loosely bound hydroxide function over there, and that hydroxide ligand can be replaced by hydride bridge between nickel and iron. So, you need this hydride bridge between nickel and iron side. So, then you need the activation, that means already we have seen that how you can cleave the heterolytic cleavage of HH bond, and the catalysis is started over there, and which is responsible for the conversion of C star in +1 oxidations to R star for hydride transfer and proton coupled electron transfer. So, this statement is very important. So, most of these cases what we find, the reaction is nothing but you have a proton coupled electron transfer reaction allowing productive binding of H2. So, how initially if we start from proton, how you get some species where your hydrogen is loosely bound to the system. That means, if it is loosely bound to the system, you can immediately get hydrogen from the system. So, that particular case, so this HH bond cleavage when it is forming, so when it is involving your nickel plus 1 oxidation step, so oxidative addition mechanism that would generate the third one, that means not C and R, X star. So, in another case it is also possible to have the corresponding intermediate, and it undergoes in this particular case 2 successive proton coupled electron transfer species to generate back the NiA C star. So, involving 3 such species we can assimilate hydrogen from the protons. So, that is the thing, that how loosely you can get. So, when your hydrogen is leaving from the system, which is eliminating from the system, that means you have loosely bound hydrogen to the system. Already the electron transfer has taken place, that means already you have transformed this from the proton and you have the loosely bound hydrogen to the system, and that hydrogen can be removed from the system. So, once we go for this binding of iron on the other side, so now we can little bit talk about the corresponding carbon monoxide dehydrogenase. So, which is a very useful reaction where we can talk about like that of our assimilation of hydrogen. It is nothing but your corresponding reversible assimilation, where we can interconvert this carbon monoxide between CO and CO2. So, this is basically a interconversion reaction what we have seen in case of H plus and H2, this is CO and CO2. So, this reaction you should remember little bit, is a very simple reaction, but we cannot reproduce it nicely in the laboratory for the assimilation of carbon monoxide and carbon dioxide. So, this giving us CO plus H2O, and it has a potential, so of minus 558 millivolt is a negative potential we can have. So, this particular reaction also, like that of our hydrogenase reaction, is a 2 electron transfer process. So, we have a reversible process which should be 2 electron transferring process. So, we have a reversible 2 electron transfer process. And now the system is little bit complicated compared to your hydrogenases. Now, we will bring some clusters, we will call them as C cluster, and also we can have what is already present there in hydrogenases also, because we are talking about large number of electrons. So, iron sulfur ferredoxin like Cuban clusters will also be there. So, involving these, and if there are some other clusters also present, we will find that. So, we find that the CODH, which is nothing but your carbon monoxide dehydrogenase. The reaction is therefore your reversible oxidation of CO to CO2, allowing anaerobic microbes to grow with CO and CO2 as the sole carbon source. So, these anaerobic microbes can take up this carbon monoxide and carbon dioxide, and your carbon monoxide is your energy supply for all these microbes. So, basically when this carbon monoxide and carbon dioxide assimilation we are talking about, we are talking something, you can have the microbial environment which is anaerobic in nature. So, this particular case, so in the early earth when you do not have oxygen in the atmosphere, your CODH was active. Along with CODH we can have another species which we will consider acetyl coenzyme M synthase. So, what we are making this? So, here the nickel site can function with that of your carbon monoxide. Now, we are talking about something which is your acetyl function. So, your acetyl function is attached to your nickel site. So, your iron site in hydrogenase was attached to carbon monoxide. If that carbon monoxide is now transferred to the nickel site, you have the CO group attached to the nickel site, and if we can have something which is the methyl function like your cyanocobalamin or methyl cobalamin. So, methyl cobalamin we know that you have the methyl group attached to the cobalt center. So, you have the corresponding acetyl function attached to the corresponding nickel site, because these reactions are very useful reactions for some of these nickel dependent enzymes. So, these nickel dependent enzymes are therefore very much important when we find the corresponding crystal structures of several of such carbon monoxide dehydrogenases. And the way we were saying that you have the C cluster, you have some other cluster. So, you have multiple metal clusters. So, one is your active site or one or more active site you can have, including 3 iron sulfur clusters, which is providing you electron to form a nickel containing C cluster at the active site. But at the active site you get the corresponding electron pool from the iron sulfur sites, and those electrons are consumed at the nickel site, and you get the corresponding catalytic transformations. So, the differences in all these cases is the ligand differences and the metal geometries of the C cluster. It is basically a different type of iron cluster. If you have a Fe4S4 cluster, and if we can have some other cluster where one of the iron site is removed, that means you have generated a vacancy within the iron sulfur cubane, and that cubane can be breached to some other metal sites, that is 4th iron site and a nickel site. So, that is basically a typical example for the different positioning of these clusters. So, you can have A cluster and the C cluster, and again one more A cluster. So, altogether here within this C cluster you can have 1, 2, 3, 4, 5, these 5, you see these 5. So, 5 clusters are there, including some C clusters. So, in these cases, that means why we are talking these two together, that CODH as well as ACS, which is acetyl coenzyme A synthase. That means, we are able to produce some acetyl function. So, acetyl function can be produced from the assimilation of the methyl group as well as carbon monoxide. So, these are also very useful reactions where we go for some different acetyl transfer reactions. So, these two basically go for 2 different types of transformations. So, this is the thing that we consider them as bifunctional proteins. So, in one closely related structure, it can function for the catalysis of corresponding conversion of carbon monoxide to carbon dioxide, that means CODH activity at one nickel iron sulfur cluster, and in another case it can function on coenzyme A to form acetyl coenzyme A at a separate nickel iron sulfur cluster, which is known as cluster A. So, you have apart from your iron sulfur cluster, you have cluster C and cluster A, nothing else. So, you have 2 clusters, one is your cluster C and another is your cluster A. So, the C cluster, when people have identified that, like that of your hydrogenases, you have iron is present, nickel is present, plus you have sulfur. So, this is basically just now I told you that if you have some vacancy from the Fe4S4 cluster. So, if you remove your one such iron center from there, that means you have created one vacancy on Fe4S4. So, simply you can draw, that is how nicely you can draw that thing, where is your vacancy. So, this is your sulfur. So, if you have sulfur over here, it is basically Fe4S4. So, now you have generated this one, that means you have Fe3S4 unit, and this Fe3S4 cluster. So, since you have generated some vacancy, so it is still a cluster, and now within that cluster we will be putting a binuclear motif, which already we have seen in case of hydrogenases. So, in the previous case where we have seen that you have separate Fe4S4 cluster, but now here you have some degradation is taking place and you have generated some vacancy, and this vacancy is attracting something and will be responsible for producing your C cluster of where the C cluster is present. We have seen just now that how much complicated your thing is, that when you see the protein structure, but the C cluster, but we are all the time we are focusing our attention on the metal center, do not worry for that. So, we are focusing on the metal based cluster. So, you have your C cluster which is present in CODH, responsible for the reduction of CO2 to CO, and now you bring this like your this particular unit, that means nickel iron site, and you have iron. So, already we know the mechanism from that nickel iron hydrogenases. So, you know that this particular motif can be stabilized very nicely by putting cysteine sulfur. So, you put cysteine sulfur, you get a bridging unit. So, once you know, that is why we are starting our discussion from the hydrogenases, that this part if you know that this particular ligand is a very useful ligand, cysteine sulfur, any thiol ligand is very much useful not only for giving you some binuclear entity, but also a heterobinuclear entity. So, your cysteine sulfur will be available there, which can be utilized for binding your iron site and this nickel site, and this site if you bring this, that means this particular unit is there if they are close enough, that means they are at the binding distance, so close to 2 angstrom. So, if this distance is close to 2 angstrom distance, this particular sulfur can function as a mu3 bridge, which is well known. So, in this particular case, so already you can recall little bit about your iron sulfur ferredoxin molecule, that what type of cubane you can have, and in that case when you have this particular sulfur attached to this, that means all these sulfur, that means which are occupying the corners of the cubes of the clusters, all the sulfurs are mu3. But when you create some vacancy, your mu3 sulfur is becoming mu2, and again when the iron is available, it is approaching to make some new bond, and that approach basically giving you some new binding, that means this sulfur already attaching to this iron for the Fe4S4 unit is showing some other binding. So, that means the entire cluster not only accommodating a new heterometal, that means the nickel, but it is functioning as a bridge for iron. So, this cysteine sulfur is there, and you have some other ligand, some loosely occupying sulfur or some from the protein chain, and this sulfur is again your cysteine sulfur, and this is your nitrogen from the histidine. So, that basically completes the entire system, which is a very useful catalytic site where you get the corresponding C cluster within the system. So, we see that you have nickel, iron and sulfur in sulfur class in this particular cluster C. So, now we know the corresponding structure, that means that you have the nickel, iron and sulfur, how the cluster C looks like. Then you have the another cluster. So, this particular part, that means the C cluster, this C cluster and this C cluster, the structure is well known to us now. And now you have one more cluster which is your iron cluster. So, this will just do not worry for that, this will continue to the next class, that this particular entity what just now I have drawn in the piece of paper. So, this is your structure, basic structure. So, this people have identified from the determination of the protein crystal structure, and you can find this particular reference initially appeared in Biochemica Biophysica Acta. So, this when people have identified that, people are puzzled initially that how you have this particular. So, this is the cluster, you have this Fe4S3 cluster, it is attracting your nickel. So, not only that nickel it is attracting, but it is bringing that corresponding cysteine sulfur, and this cysteine sulfur can establish some amount of bridging interaction with this particular this cysteine sulfur with that of our iron. That means, you have both this nickel as well as iron in close proximity, because this particular C cluster will be responsible for this very simple reaction, that means the transformation of carbon monoxide to carbon dioxide. And next day what we will see that this is a little bit complicated reaction for the transfer of this particular one. So, this is some square planar nickel base, another one molecule we will consider then again, and that particular center which is nothing but is attached to the methyl function like your methyl cobalamin. So, if you have some biological part or biological system where methyl function is attached to that particular unit and another coenzyme is coming. So, this basically the reaction between two coenzymes which is supporting these two enzyme function. So, these coenzymes are reacting in a fashion that it is basically trapping your carbon monoxide. So, in this C cluster we are getting some reaction where carbon monoxide is transferring to carbon dioxide, but in case of A cluster we basically require this particular carbon monoxide in presence of this is CH3. So, this CH3CO, that means you use this carbon monoxide for the generation of acetyl function. So, you get the corresponding acetyl coenzyme function. So, acetyl coenzyme function again will be dependent on your nickel center. So, how nickel is useful or how nickel is so useful as a catalytic site, that we will see from in our next class also. Okay, thank you.